Hello, everyone, and welcome to the 2020 MSAP Symposium, virtually given by me, Talia Smith Kelly, and I'm going to hand it off to Shahada to give you the rest.
Okay, so hi, my name is Shahaadah, and we will be talking to you guys about which sensory method gives the best resolution. Okay, so how does color space help us understand odor space? Well, what exactly is odor space? We don't really know how to describe it yet, but what we do know is that we want odor space to resemble or to be like color space. Color space is a multidimensional space where colors are mapped out by their similarities. They can be created on computers with spaces based on the RGB color model. RGB stands for red, green, and blue, and each of these colors are assigned to an axis in that multidimensional space. So like everything we see, they're made up of those three colors. We could take a picture of colors and they won't degrade, but if we try that with odors, they would. We want to create a system of odor space that does the same thing color space does, but we don't know what the dimensions and the axes of those odors are. We don't have a quantitative way of describing odors. So we're trying to figure out odor space. So our goal is that what we want to do is to map out odor space so that odors that are similar will live close together and odors that are dissimilar will live further apart in the space. We want to develop a model that predicts the description of complicated or complex odors, and by doing so, we are using several different methods. And within these methods, we want to know which one would produce the most data in telling these odors apart.
When evaluating these methods, we're trying to see which method is the best for quantifying odor quality. And there's 3 different metrics that go into it. The first one is speed. So how long does it take to complete this method? A good method is one that can give you high quality information in a short amount of time. One that gives you high quality information in a longer amount of time isn't a good method because it takes too long. The second one is consistency. So if I say this today, will I say the same thing tomorrow? So say I was given an odor and I think it smells like a red flower. Well, I believe that it smells like a red flower today, tomorrow, or in the next week? Or will I think it smells like something different? For example, a banana. And if I think it smells like a red flower, will everybody else think it smells like a red flower, or will they smell something different as well? If a method shows good consistency, then everybody should smell a red flower, and each description of an odor should be similar or near the same. And the last one is resolution. So how well can we differentiate between odors? With resolution, we want to be able to tell the small differences between odors that are described. So here's a small diagram of color from green going to yellow. With resolution, we want to be able to pick up on the small perceptual differences between odors. So the idea is that if we're able to pick up on these small perceptual differences, we're able to tell the difference between different shades of green and the way it transitions into a different color that may be yellow. And resolution was our main focus this summer, and that's how was our main metric of focus this summer.
Okay, so we have our methods. To collect our data, we use a software called RedJade, and within RedJade, we collected data for 3 methods. Each method had a different task. So during my first week, I actually participated in RedJade to do each method. I received an odor kit, and that is the odor kit right there, with 50 unique odors to describe. So the 3 methods were kata, rata, and napping. So kata, which is check all that apply. In kata, we were given 71 attributes or descriptors to choose from after smelling an odor. So if you thought the odor smelled like green or floral, maybe both, you would just check the boxes. In RATA, which is rate all that apply, it's a similar representation to kata, but instead of checking a box, you're also rating how applicable that descriptor was to the odor. So after choosing the attribute, you would just rate how much you perceive of that odor. And in napping, it's a different kind of method than RATA and kata. It's an arrangement of odors. So we give you 15 odors to smell, and then you would place the odors that smell similar close together and odors that smell dissimilar further apart. And here we have our research question, which is which method gives the best resolution, and we based our hypotheses from it. So I hypothesized that RATA will give the best resolution, which is rate all that apply. My reason is because in RATA, you can rate how applicable that descriptor is to the odor. So for example, if you have three odors, odors 1, 2, and 3, and they all smell minty to you, but odors 1 is more applicable than odor 2, and odor 2 is more applicable than 3. You would rate 1 being 5, the most applicable, 2 being in the middle, and 3 would be 1, the least applicable. So my reason for giving this example is to show that we can tell whether the method is able to describe these small perceptual differences.
My hypothesis is that napping would have the best resolution. I believe this because with napping, you use odor space based on your own perspective. And with this, you didn't have to use a set of descriptors to define whether how similar or different they were, but rather the physical space that you put them in, as well as even if there was descriptors being used, it wasn't, it was able to, like, you were able to identify some similarities and some differences, but with like just not being with not using those as like a priority for describing how the odors are different and similar. I thought it would be easier.
Okay, so in our results, we are comparing two compounds and their mixtures by distance. The compounds are brenniol and pyridine. We should see a small perceptual distance between the mixture and its most dominant compound because they're similar. And we should see a larger perceptual difference between the two single compounds because they are dissimilar. We used a PCA plot. We used the PCA plot to make maps of odor space. So PCA stands for Principal Component Analysis. A PCA plot shows clusters of samples based on similarity. So the y and x axes are the principal components, and they try to put together as much information as possible into the x-axis and put the rest into the y-axis. The percentage part of the y and x-axis are how much of the information is there. So it grabs information from many dimensions and puts it into a two-dimensional space, and it gives us an idea of how the odors in odor space could possibly be arranged. So here we have a CADA, check order to apply, PCA plot. So in CADA, it's describing the checked attributes, which are the x and y axes. The dots that you see represent an odor. So in this PCA plot, we are looking at two unique odors, two unique compounds actually, brenniol and pyridine, as well as their mixtures. The lines represent the distance from one single compound to its mixture and to the other single compound. And then we have the cation comparisons. So brenniol and pyridine should be the furthest because they are dissimilar, and brenniol to brenniol would be the closest because they are the same compound and should smell exactly alike. So we don't see the perfect trend, but the trend is there. The trend we want to see is brenniol getting closer to the mixtures and itself. So from left to right, the distances of the lines should be decreasing or getting smaller. Next we have RADA. This is a RADA PCA plot. Rate all that apply. So in RADA, it's a similar representation to CADA, but describing the ratings of the odors, which is the y and x axes. In this PCA plot, we're also looking at brenniol and pyridine and their mixtures as well. The lines also represent the distance from one single compound to its mixtures and to the other single compound. And then we have our line comparisons for RADA. Again, brenniol and pyridine should be the furthest because they are dissimilar, and brenniol to brenniol would be the closest because they are the same compound. So the trend isn't perfect, but it's definitely there. The trend we relatively want to see is brenniol getting closer to the mixtures and itself. So from left to right, the distances of the lines should be decreasing or getting smaller. And lastly, we have our napping PCA plot. In napping, it's describing the placement of the odors, which is the y and x axes. So like the other two methods, we are also comparing brenniol and pyridine as well as their mixtures. The dots represents odors. The dots that are closer together are similar. The dots that are further apart are dissimilar. In our line comparisons, so brenniol and pyridine should be the furthest because they are dissimilar, and brenniol to the 90% mixture should be the closest. You don't see the perfect trend, but it is there, just not as much as the other two methods. The trend we want to see is bringing y'all getting closer to the mixtures. So from left to right, the distances or lines should be decreasing or getting smaller. And also, if you were wondering why there isn't 100% in napping, it's because you only receive 15 odors in napping and they can only be evaluated once.
So with this mixture, with these two compounds in their mixtures, we also look at their mixture overlap. So here is a sample graph of how we expect the ideal trend to look. On the x-axis, we have our mixture overlap. 0 represents a mixture overlap of what 0 is a mixture overlap where there's no mixture overlap at all. They're two different compounds. And 100 is 100% mixture overlap is when the compounds are exactly the same. And we expect that if they're two different compounds, then the distance should be greater as the extra mixture overlap increases, the distances decrease. And on the y-axis, we see the distances between the odors. So if they're farther apart, so if the distance is larger, they're farther apart, and if the distances are smaller, they're more similar. And here's a small idea of how mixture overlap works. Since, so here is 100% borneol versus a mixture of 50-50, so 50-50 pyridine and borneol. So because there's 50% borneol in the mixture, and then there's also 50% present in the 100% borneol, then that means they have an overlap of 50% because they share that same 50%. Here is a CATA box plot of the first method that we used. So here we can see on the the percentage overlap on the x-axis and the distance in PCA space on the y-axis. You can obviously see a difference between the distance between the 0% mixture overlap and the 100% overlap. And from left to right, as we work from 100% mixture overlap down to the 0% mixture overlap, we're able to see a difference in distance, a greater difference in distance by the time we mix the 20% mixture overlap. So what does that mean? That means that CATA is able to show, so CATA resolution is, so we're not able to see a big difference in distance, or we're not able to perceptually see the differences between the mixtures until the 20% mixture overlap. So it takes a greater amount of difference within the mixtures for the differences to be perceived. And on average, it takes a 42% change in the mixture overlap for the differences to be noticeable. Here is a diagram of the second method we use, which is RADA. Here we can see that the 0% mixture overlap and the 10% and the 100% mixture overlap is greatly different. And we can also see that it takes a shorter change in the compound for there to be a perceptual difference. So as we go from the 100% mixture overlap to the 0% mixture overlap, it only takes until the 60% mixture overlap for the perceptual differences to be identified, which means that it was better. So it has a greater resolution than CATA. Also, the average percentage that you need to change within the odor composition is 27%. And lastly, we have a napping box plot showing the odor composition and the distances in space. So the difference between this and RADA and CATA is that we use coordinates to make the distances over PCA. And here you can see that there was it takes a greater, we can't clearly see a difference in we can't clearly see perceptual differences between the 90% mixture overlap and the 0% and the other mixtures that follow before it. But we are able to see the 0% mixture overlap and the 90% mixture overlap are different and are perceptually different. And the average perceptual difference from that was 48%. So in conclusion, RADA was the best at showing the small perceptual differences between odors and gave the best resolution. So here's a small diagram that was present earlier when explaining resolution. Here you can see that with RADA, it takes a smaller change in the mixture for you to tell the difference. So you'll be able to see, like, comparing like a light green to a dark green, you're able to see those small differences. Whereas with Cata and Napa, it takes larger differences in the mixture overlap for the differences to be noticed. That concludes our presentation. I would like to say thank you. And this research was supported by the Richard L. Berkman and the individual contributions to the Monell Summer Apprenticeship Program. I also like to thank mentors Emily and Kelsey and the rest of the main lab for supporting us through through the past 6 weeks.
Yep. Thank you very much.